Hello and welcome to another podcast from Odell Technology. Today we're fortunate enough to be joined by Peter Vranes from Nutromics. Peter, hello and welcome.
Great to be here, Stephen. Thank you.
Good to see you. Good, Peter. Good to see you. Now, Peter, I just wouldn't mind if you started by telling people a little bit about yourself and how you ended up where you are today, please.
Sure. So my background, Stephen, I'm a chemical engineer. I've been an entrepreneur for about 25 years. Started off in skincare and went full circle there from concept to commercialization to trade sale. After that, the sale of that last brand, I started a new skincare brand. And we were doing a clinical study looking at monitoring carotenoids in the skin. It was a skin health brand. And we're using a optical technology and got me thinking, wouldn't it be wonderful if you could measure micronutrients in the body optically and then modify your diet? Because we're all very likely deficient in certain micronutrients. It's quite important for our health. And that's where Nutromics came from. So Nutro being nutrition, omics being the science of, and that was back in 2017. But with these things, the core vision stayed the same, which is continuous monitoring of molecules in the body, but there was a lot of pivots. So that technology we had couldn't do what we were hoping. We realized people don't really care that much about micronutrients, unfortunately. So we looked at glucose, then we looked at glucose in sweat. And it wasn't until 2020 we came across this platform sensing technology from Professor Kevin Plaxco out of UCSB. He's a world leader in the biosensing field. And it was exactly what we're after. And so there was a bit of a story, but we flew to the US, had a chat, had to do that a few times, and then eventually secured that technology. And that was really the start of Nutromics, because we had that jewel in the crown, really, the thing that we were looking for, which was platform technology that can monitor any diagnostic target or drug continuously and in real time.
Okay, so can you walk us through the science behind Nutromics patch, particularly how the DNA-based aptamer sensors function in real-time biological monitoring?
Sure. So we have it's a patch, as you mentioned, and it's got small microneedles. They're about 2 to 3 millimeters, and they pierce the skin, and they give us access to what's called interstitial fluid, or the fluid just under your skin. So we have about 3 times as much interstitial fluid as we have blood. So we're bathed in the fluid. And it's the fluid that, when you get a blister, that fluid in a blister is your interstitial fluid. So it's the fluid between the cells in tissue. And it's the same fluid that's used for CGMs, continuous glucose monitors, that a lot of people, the diabetes patches, a lot of people are familiar with those. So they measure in the same fluid as well. Now, on the end of those microneedles, we put our sensor. We coat those microneedles with a sensor. And it's, as you said, it's DNA, synthetic DNA. And it mimics how our bodies measure molecules. So the way our bodies do it is through proteins that change shape in the presence of their target. That's how evolution has evolved. Now, instead of using proteins, which are quite complex, we use synthetic DNA, or aptamers, they're called. But they do effectively the same thing. They change shape in the presence of their target. And we modify them slightly so that we can measure a signal when they change shape. And basically that's how we monitor these targets in the body.
Okay, so what clinical biomarkers are you targeting initially, and how do you prioritize these particular biomarkers?
It's a great question. We're starting off with therapeutic drug monitoring of the antibiotic vancomycin. It's a very common antibiotic given to about 1 in 6 of all inpatients in US hospital. And it's for bacterial infections that cause diseases like sepsis or MRSA or cellulitis. So these are life-threatening bacterial infections. And it's really difficult to dose this drug. So as I mentioned, very commonly dosed. but about 20% of people who get it get an acute kidney injury because it's renally cleared. And about half the doses don't reach this narrow therapeutic range. So if you're outside the therapeutic range, concentration of the drug, you're not killing the bacterial infection. So very, very difficult to dose. Right now, the way they do it, they take a blood draw, goes to the lab, comes back a few hours later, an hour or more later. And so the clinician gets a single data point that's always in the review mirror. And that's one of the big problems. Our solution of a vancomycin sensor on a patch means that clinicians will have continuous real-time monitoring of that drug. So we'll be able to drastically reduce the number of acute kidney injuries, which are very expensive as well for hospitals, about $14,000 on average. But also from a workflow point of view, it's set and forget. So we take the current dosing of the drug from 5 stakeholders to dose the drug down to 2, and halve the amount of time to dose the drug, 50% reduction in FTE. So there's a whole range of really good reasons why hospitals would want to take it up. In addition, there's our health economic shows a return of investment somewhere between 5 and 15 times the cost of the patch. So there's some really big drivers for adoption for this technology. So that's just the first product.
Okay, so from a biochemical standpoint, how do you ensure specificity and the stability of the aptamer, like within the physiological environment over an extended wear period?
Yeah, it's great. So we've been in the clinic for 2 years, and we've been doing a lot of studies looking at sensitivity, selectivity, a whole range of different things. Your first point about selectivity is a really good one. So we've done a lot of studies looking at other drugs that are commonly dosed with vancomycin, making sure you don't get any false positives. One of the strengths of this technology is its specificity. So it means, really just for the audience, it means that if you want to measure vancomycin, you want to make sure that things that look like vancomycin don't get measured as well. And because we think they are vancomycin. That's what specificity is all about. You want to measure the thing that you're interested in and only the thing you're interested in. So we've done a lot of studies on looking at similar molecules to vancomycin, and we have no reactivity to those things, which is very promising. There's a reason for that. So DNA is made of these base pairs, and we start off with literally a database of 1 million billion different combinations, and we bring that down to 1. And because we start off with such a large database or a data set, it gives us an ability to be very specific to the particular target of interest. And we see that in the results.
I think so. What does continuous monitoring unlock for clinicians that a single point diagnostic can't achieve, especially in something like the acute care setting?
Yeah, so, well, for the first product, vancomycin, it results in AKIs. 20% of patients getting the AKI because it's that single data point that's delayed is grossly inadequate to enable them to dose that patient. So and that's why we get such poor outcomes. What they need is far greater data in order to adequately dose the patient and keep them in that therapeutic zone and outside the toxic range. They don't have it today. There is no technology that exists outside of what we have that can monitor a drug continuously and in real time.
Okay, so how do you envisage your platform adapting to multiplex diagnostics, measuring several analytes simultaneously?
Yeah, so that's a big limitation of in what we call enzymatic sensing. So continuous glucose monitors have been around for 20 years, and 20 years later, they're a single sensor on a single patch. And it's because multiplexing is extremely difficult with our technology. We don't have that problem. So we can multiplex. We've already got data on 2 drugs that we've multiplexed on the one device. That's a world first. And so we're going to leverage that through panels. So you think about when you go to a hospital or if you get a blood test, typically you're not getting a single analyte, you're getting a panel, because panels are what clinicians need to make informed clinical decisions. We're delivering that. So our second product is as a product for the diagnosis and treatment of sepsis. The number 1 killer in hospitals is sepsis. the biggest disease cost to hospitals is sepsis, and one of the fastest disease states is sepsis. So every hour that treatment is delayed, risk of death goes up about 7 to 8%. So it lends itself perfectly to continuous real-time monitoring. So we monitor things like lactate, and there's certain requirements for hospitals. It's called a SEP1 bundle. They have to measure certain things within a certain time, and if they don't, they get a penalty. And one of the main reasons why they get this penalty for sepsis is they don't measure lactate at the 3 and 6 hour mark because of just workflow difficulties. So there's some of the drivers of adoption for a sepsis patch. So we're having a lactate sensor that's continuously monitoring lactate means that they'll never miss another lactate measurement again, and they'll go a long way to avoiding those penalties. The number one cause of penalties for hospitals is missing this SEP1 bundle.
So how does your data acquisition and transmission architecture manage things like noise and drift or real-time calibration at the bedside or the home setting?
Yeah, so they're all big ticket items for us. Signal to noise is one of the big issues that we have to deal with. So as we develop the technology, we're always looking for ways to increase that signal, reduce the noise. As we improve the electronics, we get lower noise. As we improve the surface chemistry, the way we lay down these DNA sensors, that improves our signal. So there's like, for example, when we first went in, our first in human was back in 23. When we went back in 24, we had a 30 times improvement on our signal. And that was because of things like the surface chemistry improvements that we made. So there's a whole range of things. We've got a team of about 60 that are working on this in, as you mentioned, Melbourne and San Diego. And they're the things that we're working on to improve precision, accuracy, repeatability, calibration, the way we sterilize the needles because they're being applied into the skin, a whole range of different things to get this to market.
Do you foresee partnerships with pharma or digital therapeutic companies for companion diagnostics or drug titration support?
Yes, we do. We're deep in discussions with a number of pharma partners. This is to assist them with their drug development trials. We can speed up those trials through high fidelity PK models or pharmacokinetic models. So right now, in the preclinical space, when they're doing tests on animals, they get limited amounts of data, which means their PK modeling can be often problematic, and often drugs fail because of poor PK modeling. We can go 1000x on that amount of data by continuously monitoring the drug under development, the safety marker, and the drug efficacy marker, all multiplexed on the one device.
So is there a companion app or an AI layer involved in interpreting these continuous streams of biochemical data?
We do when it comes to the design of the DNA sensor. So we use AI as part of that process, going from that million billion combinations down to one. We're increasingly doing that more and more because it speeds up our sensor development. We have an interrogation technique. It doesn't require AI, but that's a very proprietary part of our technology stack. And moving forward, we will be getting more into predictive analytics.
Yeah.
So predicting the curve in a particular patient, but we need more data for that. So that'll be a later development that we'll be doing.
So that's your shift towards precision medicine.
Correct. Well, the ultimate for drug delivery is closed loop systems.
Okay,
so that's where. Yeah, that's where you're monitoring the drug and you're dosing the drug continuously, and you've got very, very good control when you can do that. Just like the artificial pancreas, you're monitoring glucose dosing, insulin. What we're going to see in the future is a lot of closed loops coming into the space,
Yes,
into
yes,
healthcare.
well, I agree.
Yeah.
So what's your long-term vision for Nutromics? A diagnostic platform company, a data intelligence layer, or both?
Well, it's both. I mean, I always say that the most valuable thing we'll have ultimately in the company is the world's largest molecular data set. Because we're not getting single data points when you get blood draws. We're getting longitudinal, continuous data on multiple analytes. That's never been done before. So that's going to be a source of new product development.
Yes.
I'm going down the track a bit here,
Yeah.
but when you couple that with AI, then the ability to discover early signatures of disease states that have never been discovered previously because you just didn't have those two enabling technologies becomes real.
Is there something that you're passionate about and want to talk about?
I'm passionate about helping to transform healthcare from a very reactive system that we have today to a proactive system. Now, there's nothing groundbreaking there. The question is how. How do you do that? Right now, we wait for symptoms to appear, to then go get them checked out, and then we go through the whole diagnostic process in hospital, and we are often catching acute and chronic diseases way too late, way too late. And the first thing that anyone ever says when they're diagnosed with an acute or chronic disease is,
Yeah.
gee, I wish I got that earlier.
Yeah.
Now, there are molecular signatures of these disease states in the body that we can't tap into. We don't have the technology. We don't have the ability to do that. Where we want to take this, and we're going to start in the hospital, but then we're going to move outside the hospital, and we want to be able to capture these molecular signatures at the earliest possible stage. This technology has the capability to do that, and that's exactly where we're going to take it. And that is a proactive healthcare system.
I agree. I agree. So how do you manage the challenges of signal overload, not just collecting continuous data, but making it clinically actionable and not just nice to know?
Great question. Yeah, the data for data's sake is an absolute pointless exercise. I think we've all seen that in the past, where our sensors are monitoring biomarkers that are already in guidelines. So we're not implementation is where a lot of medtech fail. Great ideas, great products, but you're asking too much of clinicians and hospital systems to change how they operate, and that's where it normally all falls down. So we're not doing that. What we're doing are known biomarkers and that are already in guidelines, and we're just making it easier for clinicians to comply with guidelines and treat patients better. And that's our low-hanging fruit. That's what we're doing.
Okay. Are there any unexpected cross-reactivities or biophysical hurdles, really, that you have to overcome with your DNA platform in vivo?
Yeah, so the cross-reactivity, that goes to the specificity, the question that you had before. If we're seeing cross-reactivity, then we're going to get false positives come up when we test things that are similar to vancomycin, for example. And so we're not seeing those. We're not in the market yet, so we're continually developing the product and testing these things. But it hasn't been something that has come up at this point. And like I said, because of the fact that we start with such a big data set, the specificity is one of the strengths of this tech.
Yeah. Okay. So is there signal decay at all? And how do you calibrate or compensate for biological variability?
So the first question is we call that drift. Is there drift?
Yeah.
So for therapeutic drug monitoring in a hospital setting, where we'll have 2-day duration. Now, 2 days is about 2 to 3 days is about the maximum that you'd want to have a needle in the skin, especially in the ICU for infection control. So they typically want to turn that over every 2 to 3 days. So we think we're comfortable there. We've got some early data actually on another drug where we've really pushed the duration, and that's for, like, say you want to monitor immunosuppressants in the community setting, if for transplant patients, for example. You might want to look at something like a 7-day patch, because the economics are much more favorable. So we've got some early data showing 7-day duration without drift. That shows that's well within the capability of technology. We've got to push that further and develop it further, but that was very encouraging. And then, sorry, your second question about biological variation. We're measuring in an individual. We're not using population data here. So where you see that becoming problematic is where, if you're dosing a drug that requires what we call AUC, area under the curve exposure, to do that today, you have to use software called Bayesian software. And Bayesian software tries to estimate what that curve is from some sparse data. And they use population data to do that. That's where individual variances become potentially problematic, because people are so different that when you use population data, it's estimation. We are a direct measurement of an individual, so we're true personalization.
Could your technology look at something like methotrexate or lithium?
We have a paper on methotrexate. In fact, lithium is another great one. There is a paper on lithium. This is some of our collaboration partners have done this. And both of those are candidates for continuous monitoring. So you're spot on there with both of those.
Okay. So what about, how far off are we from continuous biochemical tracking in oncology, where you might detect real-time changes in tumor biology or treatment response?
Oncology is one that we've looked at. There's a lot of personalization in that field. We haven't. Look, there are some drugs that our academic partners have developed using our technology for certain chemotherapy drugs. It's not one that we've gone deep into, though. We're starting with infectious diseases, because we think there's a clear and compelling need there. But certainly one that probably warrants more attention from us. There are just so many opportunities here for us to develop sensors and have a huge impact in those spaces. Unfortunately, we can't do it all. We have to be really focused, because we're a platform, and we will at one point get to those things, but we have to resist the temptation to go after a lot of different things. Yeah.
Okay. Well, I've enjoyed talking to you today, Peter. It's a fantastic endeavour that you're undertaking, and I really think it is a game changer. Tell me, regulatory affairs, how are you coping with that? Because that's a bit of a minefield for you.
It can be, yeah. So we've got a pre-sub about to go out. So we're looking at a de novo pathway for the first product, and post future products. We need to go deeper in this, but we suspect we could use as a predicate for future products and go a 510K. Yes. That's the plan at this stage, albeit we really need to go a bit deeper into that for those future products.
Have you started talking to payers at this point in time?
We do. We have a number of hospitals as well that we are very close to here in Australia, where we do clinical trials, and in the US. There's existing DRG code for sepsis that this would fall into for that first product. So that's deep into your space. So there's some things that we already have that are advantageous for that first product. But yeah, that's where we're starting to really go deeper now.
All right. Well, I wish you all the very best. It's been a pleasure talking to you today, and thank you very much for your time. It was wonderful, and I wish you all the very best of luck.
Thank you, Stephen. Appreciate it.